Welcome to another lecture. This lecture is about nitrogen cycling. So in the past, in this course, up to now, we have talked a lot about nutrients. We've talked about uptake of these nutrients through membrane transporters. And we've looked at membrane transporters on the cellular level. And we have talked about the fact that often these nutrients are taken up against their concentration gradient, and that requires some sort of energy. And the energy that often is used for that is a hydrogen or proton gradient. And that proton gradient then in turn is created by ATPases or hydrogen pumps that pump the hydrogen in and at the cost of ATP. So then using this hydrogen gradient, transporters can pick up nutrients from the environment. And so today we want to talk about nitrogen as one of those particular nutrients that plants need. So if we start by looking at the nitrogen cycle in the environment, you see that there's a circular way of nitrogen, how it flows from nitrate that can be picked up by higher plants and then they can produce amino acids, which then can be used by higher animals which eat these plants, and they will excrete the nitrogen as urea or ammonia. And then bacteria, mostly in the soil, can further metabolize this nitrogen. These are called nitrifying bacteria, and they go through nitrate, and they can then produce nitrite, and then they can go through and they can produce nitrate again, which can be picked up by plants. Now there's also all this atmospheric nitrogen that's available, and atmospheric nitrogen can be fixed by nitrogen fixing bacteria, which produce for the most part nitrate, which is available to plants, or ammonia as well. And then that nitrate again can be picked up by the plants. So the reactions that are most relevant for plants are nitrate to nitrite to ammonia, and nitrogen directly incorporated into ammonia, which then becomes ammonium, which is the form that plants can pick up. And so we're going to talk about these reactions today. So there's also the atmospheric nitrogen. That atmospheric nitrogen, that can be fixed through biological fixation, so that's bacteria and plants, but it can also be fixed through energetic reactions, so lightning and not quite as violent lightning, just light reactions from the sun can also induce chemical reactions that fix nitrogen, usually in the form of nitric acid. And lots of nitric acid in the rain or in the atmosphere creates acid rain. So we are mostly interested in these reactions today. So let's orient ourselves a little bit here in metabolism. You've seen these types of pathways now multiple times. Again, here's our glucose, glycolysis, here's the TCA cycle. And what this particular graph shows are multiple branching points within these pathways where amino acids are made. So you see that all of these orange shaded compounds are amino acids. And so there are multiple pathway branch points where amino acids can be produced from precursors such as pyruvate or phosphoglycerate or 2-oxoglutarate. And so this is the part of the citric acid cycle that we will spend some particular time on today, where oxoglutarate forms glutamate, which then can produce all these amino acids and can also be used for other reactions in the cell, which we'll come back to in a second. So nitrogen incorporation into amino acids occurs in various places in primary metabolism. All right, let's take a look at nitrate assimilation. So for nitrate assimilation, there's a specific nitrate hydrogen high affinity cotransporter. And if we go back to this figure, which you've seen before from your book, you see that it's right here. Right? So here's your nitrate hydrogen cotransporter. This is not an anti-transporter, this is a symporter. So When nitrate is picked up and added into the cytosol, there are two important enzymes that catalyze the subsequent steps. There's nitrate reductase, and then there's nitrite reductase. Nitrate reductase catalyzes the reaction in the cytosol that you see down here. So there's nitrate and NADPH, so an electron carrier. And NADPH can, for example, come from the light reaction, right? So it can be produced by photosynthesis in the light reaction. That plus a proton can produce nitrite, and then the oxidized form of NADP plus some water. And then that nitrite here is this nitrite, and that can then use use the ferredoxin in the reduced form, which will then become the oxidized form. So again, this is an electron carrier, so it's not this carrier, it's this carrier. And it adds all these hydrogens. So it's a reduction reaction that occurs here, nitrate reductase, that reduces the nitrite all the way to ammonia. So this ferredoxin here that comes from the photosynthetic light reaction, or from another pathway that we will discuss a little bit later on in the course as well. That's called the oxidative pentose phosphate pathway. You've probably not heard about that so far. So I just wanted to show you where you've seen this before, the ferredoxin. So in the light reaction, right, that is the place where ferredoxin also has a role. It's not the role that we're discussing here right now, but it's an electron carrier, just like plastoquinone and plastocyanin, and of course all the other compounds in between here that can shuttle electrons from one step to the next step. But this is a sort of universal electron carrier, just like NADPH, that can reduce other proteins or other substances. And in this case that we just discussed, it reduces the nitrite to ammonia. So that is where reduction power comes from, where electrons come from, plus these hydrogens. Okay, so once you have ammonium has to be assimilated into organic compounds. So for ammonium assimilation, I have this pathway here for you. So you can see where the ammonium goes into the carbon skeletons. So it starts with glutamate as an acceptor and ammonium. And then ATP is required, as well as an important enzyme that's called glutamine synthase. So glutamine synthase and ATP, together with ammonium and glutamate, produce glutamine. And so the ammonium right here is now an amino group over here. ADP is produced, energy is used up. That glutamine cannot become glutamate or amino acids unless it reacts with a 2-oxoglutarate. And so that 2-oxoglutarate is a product that is made in the TCA cycle. So this plus this will produce these two compounds over here. Now these two compounds over here are the same, they're both glutamates, but you can see that the ammonium here in this reaction, which is catalyzed by the enzyme glutamate synthase, or GOGAT, that uses NADH plus H+, so another electron carrier, or it can use reduced ferredoxin, and it will add a hydrogen to these two compounds to produce these two compounds, and then move the amino group here from here to here. So in the end, what you end up with is two identical compounds, two molecules that are both glutamate, and you make those from glutamine and 2-oxoglutarate. And just to show you again where we are, we're down here in this area of this pathway. So why am I bringing back the photorespiration pathway? Well, that's just so that you can see that glutamate and 2-oxoglutarate are not just used for making amino acids. They also help in a number of other metabolic reactions, and some of those you have already seen. So if you remember, in the photorespiration pathway, we usually, for the most part, spend our energy on looking on the outside reactions, right, for the salvage reaction so that we could produce a 3-phosphoglycerate again. But all these inner reactions where electrons are shuttled and where the 4 carbons become 3 carbons with the loss of the carbon dioxide are dependent on these reactions on the inside of this pathway. And if you look closely, this is all glutamate or 2-oxoglutarate. So that is where these substances also play a role. So just a side note, this is not what we're focusing on right now, but I do want you to make these connections as we think about substances in the cell. Okay, so let's just put together what I just told you. It's just a summary reaction that you used ammonia. It requires glutamine synthase. It requires glutamate and ATP. You're producing glutamine. And then in the next reaction, you're producing glutamate. And here are our glutamates, which then can be either used for certain reactions in the cell or for the production of amino acids. So this is another way. You can pause the lecture here for a second if you want and just sort of see where we are in the cell with all these different pathways. We'll come back to that, so don't worry about it right now. But here is sucrose, here's glycolysis, here's our TCA cycle, and then again here is oxoglutarate and glutamate producing proteins, other amino acids, or a number of other organic compounds that are important in the cell as well. So you will see these types of graphs more later on. Okay, so let's move on to the bacterial fixation of nitrogen. So plants can also form symbiosis with nitrogen-fixing bacteria in exchange for carbohydrates. So the bacteria get the carbohydrates, and the plants get the nitrogen. And so they get it in the form of the nitrate or ammonia, and the bacteria then, of course, get the carbohydrates. And so the reaction that's going on here is a different reaction. It happens in the bacteria, and it uses a different enzyme. And it uses an enzyme that's called nitrogenase. And so in this nitrogenase reaction, atmospheric nitrogen is fixed into ammonia, and it costs 16 ATP. So it's a very energetically costly reaction. Electrons and hydrogens plus all this ATP to produce the 2 ammonia molecules here. And then subsequently, just through dissociation and pH-dependent reactions, we can get an equilibrium between ammonia and ammonium. Okay, so how does the interaction between the plant and the bacteria occur so that this symbiosis can form? So it starts with the plant exuding flavonoids. Flavonoids are compounds, we'll talk more about those later on, organic molecules, relatively small organic molecules that attract rhizobia bacteria. So once rhizobia detect flavonoids in the soil, that compound activates a certain gene in the bacteria. And so this is a bacterial NOD-D gene. There's a number of NOD genes in nitrogen-fixing bacteria. And once NOD-D is turned on, other NOD genes in the bacteria are also turned on. And then the bacteria produce these NOD factors, so it's basically the proteins that are made from the NOD genes. So you can see here that there's a root, and we have a root hair, and there's exuding of flavonoids going on. And so the bacteria are starting to be attracted because of that interaction between the flavonoids and then the production of these NOD factors. And so then when these NOD factors are made, plants can react to these specific NOD factors, and they then in turn produce another compound. And so these compounds that the plants now produce are called lectins. These are sugar-binding proteins. So lectin is sort of a general term for all kinds of sugar binding proteins. And they then direct the bacteria directly to the root. So if they are sort of on the outside of the plant, they now find their way where they're supposed to go. Bacteria are then attached to the root hairs, and then there's some more nod factors that are induced and that are induced in the bacteria, which then induce this growth response of the root hair, and this is called curling or root hair curling. It sort of engulfs the bacteria. And then after these bacteria are engulfed and enter the root hair, they create something that's called an infection thread. And after entering the cell, the plant cell, they can then fuse with the cell membrane, and then from there they can actually infect a number of cells inside the root. And so from there, with that, they can then transfer the ammonia that they make, the ammonium that they make, directly to the plant. So it's an intricate dance that goes back and forth between the plant cell and the bacterium, and then back to the plant cell and back to the bacterium, with all these factors that are exuded and recognized so that certain reactions can occur. Here's just a picture of what such interaction or symbiosis looks like. You have the roots, and you have these nodules that are made by the plant that are housing the bacteria, and then the infection threads go all the way into the roots. This is an example of a interaction between bacteria and a bean plant. And then I just have a little table here for those of you interested to see which kinds of plants are interacting with nitrogen-fixing bacteria, because that's not very many. It's actually only a relatively small part of the plant world that has this symbiosis. And so they act directly with certain specific nitrogen-fixing symbionts. And again, from among all those bacteria, there's only a relatively small number that can do the nitrogen fixing. There's some free-living nitrogen-fixing bacteria as well, but these are the ones that are interacting with host plants. Great, so that's it for today. I'll see you in class.